Hi Beckman students, AP Bio students. Today we're going to be talking all about the light reactions. So let's go ahead and get into it. First questions we have here are where do the light reactions happen, and also what are the reactants and products, really meant to set the stage so that later on we can kind of fill in the details. So again, quick review, where does it happen? It happens in the thylakoids. As you can see in this chloroplast over here, the thylakoid are these green sacs inside of the chloroplast. And so that's where the light reactions occur. The reactants are light, water, NADP plus, and ADP. Those are all the arrows kind of pointing into the light reaction. You can also say phosphate. And then the products are oxygen. So I know it's blocked right now, but the oxygen is coming out of the light reaction. So this is where we got our oxygen that we breathe, that's created by our plants. We're also making a little bit of ATP and some NADPH, and that's going to be very important later on in the Calvin cycle. So obviously light is an important part of the light reactions, hence the name light reactions. So the question is, what happens to a chlorophyll molecule? Chlorophyll is a pigment that's found inside of thylakoids, so we'll talk about that later on, when it's exposed to light. Well, as you can see here in this diagram, the light is absorbed by the electrons of the chlorophyll molecule. It's going to excite those electrons, and they're going to be raised up to an excited state. But electrons don't like to stay at this excited state. It likes to fall back down to its ground original state. And so as it falls back down to its ground state, it's going to release some heat. It can also release some photons. In this case, the example is fluorescence. So the next thing I had your classmates do was to label the photosystem. So if you haven't already done so, make sure you pause this video and you can try to label it on your own using the word bank to your left. But I'm going to go ahead and get into the answers of how you can label it. So first, I'm going to just set the stage of what we're even looking at, where in the chloroplast we are. So this membrane, as you can see, the phospholipid bilayer, here's the thylakoid membrane. That's where you find your photosystems. The light gray outside is the stroma, and the darker gray inside is the thylakoid space. So again, just keep these locations in mind. They're super important, especially later on when we talk about the electron transport chain. So first, you're going to have a photon hitting one of these pigment molecules. These are the green dots found inside of the light harvesting complex. And then as you can see, from one pigment molecule to the next, there's a transfer of energy. Those are the black arrows pointing from one pigment to the next. And so what's happening is, as an electron's getting excited, it's going to fall back to its ground state, release some energy, and that energy will be picked up by the next pigment molecule. And it's kind of like a domino effect of energy transfer. Finally, the energy is going to be transferred to a special pair of chlorophyll A molecules that's found inside of the reaction center complex, along with a primary electron acceptor. And so once that special pair of chlorophyll A molecules is excited, those electrons from that special pair of chlorophyll A molecules is going to jump up to the primary electron acceptor, and then after that, it's going to fall down electron transport chain. So let's talk a little bit more about the light harvesting complexes, reaction center complex, and primary electron acceptor. But after we specify the location of this photosystems, remember the photosystems are found in the thylakoid membrane. So the light harvesting complexes are, obviously they're harvesting light, and they're harvesting light using pigments. And the pigments absorb light, and we saw last week that pigments, various pigments absorb different types of wavelengths of light. So then there's the reaction center complex, which is in the middle right here. This is a group of organized proteins that hold that special pair of chlorophyll A molecules, and also contains a primary electron acceptor, which is a molecule that is capable of, as you can guess, accepting electrons, and therefore if it's accepting electrons, it's gaining electrons and it is being reduced. And so obviously the photosystems participate in redox reactions. So let's talk more specifically about the steps of the light reactions. So where does light go and what does it impact? Well, the light is going to be collected by the photosystems, as we saw earlier, and let's just put all of those pieces together. And so it's kind of backwards, but we actually start with photosystem 2. I think they were named based off of when they were discovered, and so photosystem 2 was discovered after photosystem 1. And so what happens is you have light hitting pigment molecules inside of photosystem 2, and there's going to be a transfer of energy from one pigment to the next until we reach that special pair of chlorophyll A molecules. In this case, it's P680, which is referring to the specific wavelength that it really likes to absorb. And so once the energy is transferred to that special pair of chlorophyll A atoms, the electron is going to be excited to the primary electron acceptor. And now it's missing some electrons, and so what's going to happen is we're going to split water, which is one of our reactants of photosynthesis, into H plus oxygen, and we're going to strip it of some electrons. So water is the provider of electrons for our photosystems and for our photosynthesis. Okay? So I just want to remind you that the excitation of the electrons and the transfer of energy from one pigment molecule to the next occurs through the process we talked about earlier, where a photon excites the electron and the electron falls down, and as it falls down, it releases some energy in the form of heat, and then also just energy in general. So it could be a photon, could just be energy that's being transferred from one pigment molecule to the next. So after the primary electron acceptor has electrons, those electrons are going to fall down the electron transport chain, and in this first electron transport chain, it actually creates some ATP. And so that is energy that's going to be used later on in the Calvin cycle. Then we arrive at photosystem 1, and so the electrons fall down the electron transport chain, and they end up in the special pair of chlorophyll A molecules. This is known as P700. But before those electrons get excited, there has to be light that is harvested by our pigment molecules inside of photosystem 1, and there's going to be a transfer of energy from one pigment molecule to the next until they get excited, or the electrons in P700 get excited, and they move up to a primary electron acceptor in PS1. So after those electrons are in the primary electron acceptor of photosystem 1, they can fall down that second electron transport chain, which is where NADPH is created, and NADPH along with ATP are going to travel to the Calvin cycle. And so this is just kind of like a cool analogy to help you think about what's happening to the electrons. So a photon is going to excite the electron, and then the electrons are going to fall down an electron transport chain. In this first one, we make some ATP. And then there's going to be more light that excites the electron again, and then there's going to be an electron carrier that brings the electrons to the Calvin cycle. So let's do a little quick review and label. Well, I'll just label the photosystems first. So the one on the left, the first photosystem is photosystem 2, and then the one on the right, the second photosystem that the electrons reach, is photosystem 1. So the first step is light is going to hit the pigment molecules. Again, these pigment molecules are found in the light harvesting complex. And then we're going to have some water split into hydrogen ions, oxygen, and electrons. And the reason we need to provide electrons is because as light hits the pigment molecules, energy is being transferred from one pigment molecule to the next until the electrons of P680 are getting excited and going to leave P680, leaving it empty. As they go to the primary electron acceptor. And so now that it's empty, we need to replace it with some more electrons, and water is providing those electrons for us. So then the electrons are going to travel down an electron transport chain, and they're going to create some ATP. And then in photosystem 1, similar process, we have light photons hitting these pigment molecules, exciting electrons in one pigment to the next until they excite the electrons in P700. And so the electron is going to jump up to the primary electron acceptor, and it's going to travel down another electron transport chain. In this electron transport chain, we have NADP plus reductase is going to change NADP plus, add a hydrogen ion to it. It's going to reduce, which means it's going to cause NADP plus to gain electron. Remember, Leo the lion goes grr, gain of electron is reduction. And it's going to create NADPH. And so ATP and NADPH are going to be forms of energy that are going to be transferred to the Calvin cycle. So here it is in its totality. Steps 1, 2, and 3 are just showing you where the products that we talked about earlier of the light reactions are being created. So in step 1, we are creating oxygen through the splitting of water into oxygen and hydrogen. And that's going to provide electrons to photosystem 2. In step 2 is when we're making some ATP through that first electron transport chain. And in step 3, we're making NADPH, which is another electron carrier. It's going to carry electrons to the Calvin cycle. So if you look at this picture from before, it kind of reminds you, of this, oxidative phosphorylation in cellular respiration. As you can see, both of these processes have ATP synthase. They both have electron transport chains. So I'll go back to this one, right? Here's your ATP synthase, here's electron transport chain. They're both occurring across the membrane. And so let's take a look at this diagram here. I love this, and I highly recommend that you study this diagram and understand the differences and similarities between oxidative phosphorylation and the photosystems of chloroplasts. So as you can see, both of these processes generate ATP through chemiosmosis, which is when we have an electron transport chain pumping hydrogen ions across a membrane, and those hydrogen ions are going to want to diffuse through an ATP synthase, which creates ATP. Both of these electron transport chains, whether they're in the mitochondria or the chloroplast, they occur through redox reactions, the transfer of electrons, and then they both have ATP synthases, okay, which again allow for H plus to diffuse down its concentration gradient. And also, we haven't talked about this extensively, but the electron carriers in the electron transport chains of both processes are very similar. Let's talk about some differences. So one has to do with the electrons and where they're coming from. So in photosynthesis, the electrons are coming from water and provided by water, but in cellular respiration, the electrons are provided from the breakdown of glucose. In photosynthesis, we don't need glucose to make ATP. ATP actually is used to make glucose. In cellular respiration, it's a little bit backwards, where we need glucose to make the ATP. And then finally, over here, we can actually refer back to the diagram and see where the H plus is being pumped from and into. So in photosynthesis, the hydrogen ions are pumped from the stroma to the thylakoid space, but in cellular respiration, the hydrogen ions are pumped from the matrix to the intramembrane space. So if we go back here, right, so chloroplast, we're pumping hydrogen ions from the stroma, light gray, to the thylakoid space, and then they diffuse back into the stroma. In the mitochondria, we're pumping hydrogen ions from the matrix into the intramembrane space, and they diffuse back into the matrix. So location-wise, it's a little bit different. Okay, so that is all about linear light reaction or non-cyclic light reactions. Now let's talk about the cyclic light reactions. So this is a diagram showing you what the cyclic light reactions look like. As you can see, it only uses photosystem 1, and it happens in, as you guessed it, a cyclical way. So again, it only uses photosystem 1. The way that electrons move, they go from ferredoxin, which is this FD, then they go to a cytochrome complex, and then they go to plastocyanin, back to P700, and then the electrons get excited, go back to the primary acceptor, and they repeat this cycle over and over again. And now the benefit of that is I can create ATP, but just know it does not make NADPH, and it does not create O2 either. Okay, this is an evolutionary relic because it's a way to create ATP without going through this complex process of having two photosystems. So it's an evolutionary relic in that way. Alrighty, everybody, thank you so much for watching. I hope that was helpful. And always remember, this is fine, and I can do it. I'll see you in class. Bye.